Everybody can see.
All right, let's get started. So I'm Jason Irwin. I am Logan's committee chair, so I get the pleasure of introducing him. So he was here, he came after doing his blue-collar job for a while. He was a tow truck driver for 6 years. Logan came back to school. This is one of the things I love about Central, is we attract students who come from all walks of life. Some of them say, hey, I need to go get that degree. I love this stuff. And so it's exciting for me that he decided that he loved it enough to go on and do a master's. And he picked this topic. I didn't tell him what he was going to study. It's totally his idea. In fact, I kind of just tried to dissuade him. I don't know anything about Osmia. What are you talking about? But he ran with it. So we're excited to have him here today and be able to have him defend his research. So I just want to welcome everyone. The way this works is that Logan gives a fantastic presentation, at least you better, and then you get a chance to ask him tough questions. So while you're sitting there, think of some really great tough questions to ask him. And then we'll ask you to leave, and the committee, which is myself and Dr. Lucy Botcher and Dr. Bill Kemp, who's joining us by phone, we'll get to ask him some tough questions. We're really excited to have Dr. Kemp on board. He is recently retired from the Agricultural Research Service lab in North Dakota, in Fargo, North Dakota. And he really is one of the world's experts on Osmia, among other species, including diapause regulation, which is what Logan did his work on. So I should probably just stop talking and let Logan take over. So please join me in welcoming Logan Kral. Let's go.
Thanks for coming out, everybody. Like Jason said, my name is Logan Kral, and the topic of my thesis is on cold tolerance, temperature-mediated discontinuous gas exchange, and emergence of the blue orchard mason bee, Osmia lignaria. You see right there.
Down button.
Oh, down button. There we go. Other one. So, first a little bit of background about the bees. Wild bees are becoming endangered. We had the red bumblebee, Bombus rufa, go onto the endangered species list last year. And 44% of our commercial honeybees here in the United States suffered from colony collapse. In Washington State, 100% of our commercial cherry, peach, and pear crop, as well as 70% of our apple pollination, is performed by honeybee rental using Apis mellifera, the European honeybee. And because of these problems that they're facing, alternatives are going to be needed here in the very near future to support our agricultural needs. Enter the blue orchard mason bee. The blue orchard mason bee, Osmia lignaria, is a native northwestern pollinator, endemic here to the Pacific Northwest. It pollinates 9 times the amount that honeybees can pollinate per bee. This means that you need fewer bees to cover a larger pollination area, and they're also very, very easy to maintain on site, needing nothing more than a small nesting block and a source of clay to build their nests with. They're the pollinators of many types of orchard trees, some of which we have the products of right here on the snack table. Stuff like prunus, malus, pyrus, rides, and taraxacum. That would be the prune, apple, pear, currants, and dandelions, which you see there. So, a little bit about the mason bee life cycle is it starts off in the beginning of spring, when the bees emerge out of their cocoons, when 14 degrees C average daily temperature is achieved, and at that point they start emerging, flying around, mating, and begin making nests of their own. They will lay from April to about July, at which point they will finish laying, and the eggs that they have laid will begin consuming the pollen mass, the pollen and nectar mass that is provided to them, that they foraged by their parents. So during that time, this is basically a really critical development period where they go through a series of molts from first instar through the fifth instar. at which point they then spin a cocoon around themselves and pupate. During their pupation, they then go through a very short summer diapause, kind of timed to coincide with the hottest period of the year. And after this, they then emerge from their pupal carapace and tuck it kind of down towards their abdomen, still inside their cocoon, however. And as an adult bee, they then begin preparing themselves for a winter diapause, which then sets in around September. They then, once achieving diapause in September to the beginning of October, they then spend the remainder of the year, all the way until the following spring, inside this cocoon as an overwintering bee. And here's a photo here of what the nests often will look like. They typically are beetle borers or lake reeds, some sort of a small, short tube, around 5 to 9 millimeters in diameter, and plugged with a clay cap in the end. So diapause, if you've never heard of it, is a period of it's a period of suspended metabolic activity. Essentially, it's where cellular development stops and nervous system slows down, respiration slows down. Here in Bosch 2010, they plotted the O2 and CO2 respiration of diapausing bees as they closed, and then drop their respiration down to very, very low levels in preparation of winter diapause. When temperatures get down below 14 and into 4 degrees C here, they then enter into their wintering state, complete their diapause, and then slowly begin ramping up their respiration until they reach the spring. During this time, this is referred to as their post-diapause quiescence. And quiescence is essentially a state where the bee becomes responsive to a token stimulus, which will trigger them to emerge from their cocoon and resume development and begin laying, completing the life cycle. So, Osmia lignaria go through 2 obligate diapauses per year. They're a univoltine bee, which means they do need to complete this diapause in order to complete their life cycle and reproduce. Their short summer diapause occurs, like I said, kind of during the hottest time of the year. The longer winter diapause occurs during the coldest time of the year. And both of these are speculated to be as a result of natural selection to avoid stressors during these hot and cold periods. If they didn't diapause, they would probably experience some pretty drastic reductions in their survival. So, what we know so far is that prepupal duration and temperature have a strong effect on advancing development during the summer. But that an extended warm post-pupal temperature can actually inhibit diapause and negatively affect the development of the bee. We also know that respiration rates gradually increase until closure, and then as fall sets in, and then fall back down as diapause sets in. And we know that maximum fitness can be found when the bees are held at 4 degrees C. We also know that emergence can be induced once they receive a 14 degrees C average daily temperature. And that is their post-diapause, during their post-diapause quiescence, that's the stimulus that they need to come out of their cocoons. So my questions were, are low temperatures involved in the initiation of winter diapause? Something which hasn't been looked into much yet. I also was looking for metabolic responses in relation to testing temperature, and what effect pre-wintering temperatures have on emergence. And additionally, I also sought to clarify what their level of cold tolerance was. Previous studies have looked into the cold tolerance in Osmia from Nova Scotia or Utah, but so far Washington and Lignaria have not been evaluated. The nests that I sourced my bees from, which all came from a commercial supplier called Crown Bees of Woodinville, Washington, are all from a source of mixed wild and previously released parentage in mixed tree fruit orchards in the Puget Sound region here, ranging from as far out as Sultan to Snohomish, down into Duval, and across the Sound over on Bainbridge, Kitsap Peninsula, and as far north as almost up into Squid, Port Townsend. And I distributed my bees thus. So I had around 400 bees that I used in total for all of my experiments. I selected out 168 bees to use in my early winter metabolic experiment. Equal numbers of males and females, 42 each. And I brought them out and exposed them to their temperature treatments on the 5th of September, and then set aside another 84 bees on the 22nd of September of 2017 to use as backups in case there was any problems, if we had any unexpected deaths or anything. These bees were then emerged the following spring on February 27th, 2018, and I emerged I tried to emerge all 168 bees that I had available to me there. I then did a effective testing temperature on metabolic rate experiment, which lasted from November 14th until November 29th. That should actually, my goodness, that should be actually 2017 there. Sorry about that, little typo. That was 70 random bees. And then I did a temperature of crystallization test using 60 more bees that I tested in October, November, January, and April. So that's from 2017 to 2018. The early winter metabolic experiment was arranged thusly, where I took from my bees that I obtained on August 16th of 2017, held them at around 23.5 degrees C, and then distributed them into 3 control groups and 3 treatment groups. My control groups were 22 degrees C constant control and 2 14 degrees C constant controls, one of which had a 12-hour light and dark photo period. Of these, I had 7 females that I was testing in each group. Previously, females have been shown to not differ much as far as respiration goes between female and male. So I was essentially following along the methods of prior experiments here. I also had 3 treatment groups in a 12-hour thermal period cycle. So ranging from 14 degree during the day down to 10 degree, 5 degree, and 0 degrees at night. And again, these were all with 7 females for each group. And I would sample respirometry from them every 3 days from the 5th of September 2017 until the 7th of October 2017, at which point I put them all into wintering conditions at 4 degrees C constant temperature. All of these bees that were used in this experiment were then emerged the following spring. So, the respirometry, which you see in this wonderful setup we have here, was conducted with a Lycor LI6251 CO2 analyzer, set to flow-through mode that flowed at 100 milliliters per minute, and used a airstream that was scrubbed of carbon dioxide and water vapor. Only females were used, which was the same method that had been used in Kemp et al. 2004. And this was done after, respirometry was collected after a 60-minute acclimation period, meaning that I let the bees sit at that temperature for an hour before conducting any sort of metabolic testing with them. In my early metabolic rate experiment, which had an N of 42, I did sampling for 5 minutes per chamber, again using flow-through respirometry. And then for my effective testing temperature experiment, I had an N of 70 there, and I sampled for 60 minutes per chamber. And I tested at 5 degree increments there from 0 through 20 degrees C. Right here we see one of the chambers, an example of a little device that I use to actually collect their CO2. So what I found during my early metabolic rate experiment was that 5 minutes of sampling was insufficient using flow-through respirometry. And when I tested them for longer periods of time, like 4 hours, I discovered that they exhibit something we call discontinuous gas exchange, which essentially means, these are some sample graphs here of different temperatures. I don't know if you can read that. That says 0 degrees C, this is 10, and that's 20. And at 0 degrees C, you can see that they breathe 1 time in 4 hours. At 10 degrees C, they'll breathe a few more times, and at 20, you're going to get pretty regular breaths. This is the essence of discontinuous gas exchange. It was described by Logan in 1996 as a respiration sequence consisting of open, fluttering, and closed phases. In the model that I developed off of this, respiration rate was found to be a function of body mass, test temperature, and the bursts of CO2 activity that we saw. And that had an AIC of, I used stepwise regression to get an AIC of negative 86.65 there. Of those different predictor variables, temperature was the most significant predictor with a very strong p-value and a r-squared of 0.86. So that shows essentially that 86% of the data of the variability can be explained by just temperature. Body mass and the cycling rate were also included in the model, but were not significant on their own. However, when I took them out of the model, the AIC rose, so I felt it was best to keep it. So the results of the respirometry essentially showed that when we measured them for 60 minutes, respiration rate rose exponentially. And it ranged from 6.54 microliters per gram hour of B up to 177.72 microliters per gram hour. I then also did a log transform of the data, which showed a pretty strong linearity there. When I looked at the cycling rate for these temperatures, I also saw a similar exponential relationship. Unfortunately, this was not normally distributed, so I had to use a non-parametric test. I used Cresswell-Wallace in this case. And we again found a very strong p-value with a r-squared of 0.75. So, what does that mean? Essentially, it shows you that respiration rate using flow-through respirometry were much, much lower than previous research has shown, by almost 2 orders of magnitude, sometimes almost 3. This is likely just the result of simple gas diffusion through the body of the insect rather than actual breathing. And therefore, we can conclude that using flow-through respirometry, 5 minutes of sampling is just insufficient to properly capture the respiration rate, the true metabolic rates. And if you want to use flow-through respirometry, you'll need to sample for many hours. We also saw that quantitative evidence now exists for discontinuous gas exchange in Osmia lignaria, previously unseen in Osmia and only qualitatively seen in prior studies using other megakilets. The periodic bursts of CO2 activity that we saw were performed, while performed in respirometry, were a great example of classical DGE, described by Logan. Metabolic rates ranged from 6.54 microliters up to 177.72 microliters at 20 degrees C. And the cycling rate ranged from less than 2 breathing cycles per hour, temperatures below 10 degrees C, to more than 15 cycles per hour at 20 degrees C. So after the metabolic rate experiments, then I did an emergence experiment as a kind of method to determine post hoc if any of my thermal period treatments had any effect on the fitness of these bees. And I He was able to emerge 126 bees total. The remainder of 40 some odd bees failed to emerge. And I used ventilated glass tubes, like you see here, using a 2-dram vial with a cotton ball stuck at the end to keep them from flying away. And I put them into an incubator set at a constant 20 degrees C with no photoperiod. The date of emergence was recorded, as well as the number of days they survived until they expired, starvation. Photoperiod had no effect on emergence or post-emergence vigor, and the duration of pre-wintering treatment, whether they were started on the 5th of September versus the 22nd of September, also had no effect on emergence, vigor, or body mass. So for that reason, I combined the data sets to conduct a non-parametric analysis. And the results of that analysis are here. And we can see that the group that was exposed to 10 degrees C at night emerged around 2 days sooner than the other groups, significantly so compared to the 5 degree C group and the 22 degree C group. The average time of emergence for the bees exposed to a 10 degree C night thermo treatment were emerging around 5.8 days on average, and that was confirmed with a crustal Wallace that had very strong significance and a chi-square of 17.4. I also looked at post-emergence vigor and found that there was no significant differences here between any of the treatment groups as well as controls. And the conclusions from that we can see are that warmer temperatures during the winter and the spring can advance emergence at the expense of fat body depletion, but that an intermediate thermal period that was used in this study actually was statistically significant at reducing emergence time. The 22 degree C control group, which had basically been held to a constant 22 degree C temperature all the way until they were timed to go into winter in October, they emerged longer, significantly longer than all the other groups. This partially disagrees with the findings from prior studies, which showed that longer summers would increase degree day accumulation and allow for a faster emergence. And this could be just the result of low N or potentially some other effect. Post-emergence vigor was not significantly altered by temperature treatments, and the gist of this is that we can probably conclude there is a need for an intermediate thermal period between the summer and the winter in order to successfully and most expeditiously emerge the insect out in time for early winter blooms. So then I did a cold tolerance experiment as well to find out what the supercooling point was of these bees, which had never been looked into before for Washington lignaria, although it had been looked into for Utah and Nova Scotian Osmia. I used 15 bees each time I tested, and I put them into a little 2 milliliter centrifuge vial. I dissect them out of their cocoon and then put them into the vial with an open cell foam to inhibit movement, along with a type T thermocouple placed against the sternum of the insect. These I then submerged into a NestLab ethanol cold bath, started at 4 degrees C and then cooled down by 1 degree C per minute, all the way down to negative 40 C. I tested them on October 24th to the 28th, November 27th to the 29th of 2017, January 2nd, and April 15 of 2018. An example of what it looks like when you collect this data is right here. After testing, I then took the insects out and then dried them in a 40 degree C oven to constant mass so that I could get their dry weight as well as water content. And what we saw here is that the average temperature of crystallization, the supercooling point of these bees of Washington Osmia lignaria, is negative 26.4 degrees C, plus or minus 0.6. And that is quite a bit colder than prior literature has shown. I also saw using stepwise regression that body mass was the single best predicting variable of supercooling point of these bees. And the model that I developed had a very strong significance with a P of 0.007, although the R squared of the linear model was only 0.1033. You can see there that it's got a pretty strong negative trend. As body mass increases here, supercooling point goes down. This kind of runs counter to prior literature, which has shown that typically cold tolerance is inversely proportional to body mass in Hahn, 2008. But bees from Washington and Utah are phenotypically similar in their development cycle, so I expected that we would see pretty similar supercooling points out of them as well. However, our Washington lignaria was well below that, negative 26.4 C, well below that of the Utah lignaria in Sheffield 2008, which supercooled to negative 18.4 degrees C. However, the low R squared of the model I developed shows that there's probably another factor affecting their temperature of crystallization here. Could be osmotic concentration, could be a geographic adaptation, could be their diet, it could be thermal hysteresis, or even fat body depletion. And I would, so that concludes my research, and I would like to thank my graduate committee, Drs. Jason Irwin, Lucy Botcher, and William Kemp, Dave Hunter of Crown Bees, Woodinville, Washington, for providing all the bees that I used in my study, the School of Graduate Studies and the faculty of the Central Washington University Biology Department, as well as my fellow graduate students, undergraduate assistants, and my family and friends for their help and support. Does anyone have any questions? Yes?
So in your last set of experiments that you were describing, the cold tolerance,
Yes.
why did you choose the 1 degree per minute for the cooling? Is that just by the equipment or from earlier work? If you changed that, do you think you would see a difference in the insect response?
So that is what's been used in prior studies, and that is also the limitations of the equipment. Coincidentally, actually, it's not even a linear 1 degree C per minute. As you get colder in the machine, it actually starts cooling slower. But I would not expect that to have too much of a difference, although I am curious to know, I'm curious to explore the potential for cold holding these bees in potentially a future experiment where we could see how long they could survive at a low temperature. We've seen in some prior studies that they do have a capacity for withstanding these low temperatures without any lethality, but that if held past that time, then it could become lethal.
And you don't think that maybe they could get colder if it was slower? I mean, I don't know. I'm just
Yeah, I haven't seen any data to suggest that could be the case, but I'm not going to rule it out.
I mean, in nature, when it gets cold, how quickly or slowly does it get cold?
Slower than 1 degree C
Yeah.
per minute.
Uh-huh.
Yeah,
Yeah.
definitely.
Okay.
Yeah. Yes, that.
So unless I missed something, it looks like you didn't group them together in a vial for the flow through respirometry, right? You were doing single bees?
Yeah, individual bees per vial.
Right, so just if you're getting such really low metabolic rates, or you're expecting pretty low metabolic rates, would you consider, putting them together in a group to get like a group average per se?
Yeah, that could potentially be done if I was kind of scaling back and looking at more of just trying to establish a kind of a general metabolic rate, I suppose. But then it would be really difficult to try to normalize on the per gram of bee because they'd all be different weights at that point. So it might muddy the results. So most of the testing on respirometry with these insects has been done on a per individual bee basis. I was basically just piggybacking on the methods of prior research here. Yeah. but it's possible. Yes?
So you originally hypothesized that a cooler temperature regime would result in accelerated dipause.
Yeah.
But it was actually the medium. Can
Yes.
you summarize to us why? Why is that the case?
You know, that's a good question. I would imagine that going to too cold of a temp too quickly is just, it doesn't give them enough time to properly allocate any cryoprotectants they may have, or maybe it just might be too sudden of a change for them, I would imagine. But I don't know. That's a great idea. Good idea for further.
Have others found the same thing?
What's that?
Have others found the same thing?
No one else has looked for a low temperature cue with these bees before. They've explored high temperatures and what effect longer summer temperatures would be, as well as wintering temperatures. But during the period between summer and winter, no one has really looked at what effect a low temperature, if there was a low temperature which had an effect. So this was kind of virgin territory, so to speak. So, yeah. Yes, sir?
What was the viability of bees that were exposed to 10 degrees Celsius as opposed to a 22 in the emergent stage? Did you follow that through, or did you have to kill them?
Yeah, no, well, they all eventually died. That's how I was able to figure out how.
At different rates.
Yeah, that's how I was able to figure out if there was a difference in the post-emergence vigor of these different thermal treatments. And I didn't see any differences as far as their survivability after emerging from thermal period to thermal period. It seemed to be they're relatively unaffected. The 22 degree C control had a very slightly lower post-emergence vigor than the other thermal period treatments, probably as a result of fat body depletion, so they just didn't have enough energy come the following spring. So then they would burn through their fat stores and then die either while trying to emerge or before they were able to chew their way out of the cocoon. But I did see some differences in survivability, but it was not because of the thermal periods. It was actually because of the, where's it at? Nests. So I did see that there was some differences here with the dark line being the number who died and the gray colored section being their survival. But not from thermal periods, unfortunately. That seemed to have no effect.
So by nest, you mean the different sites that were collected from?
Exactly. Exactly. Yeah, the different nests they came from. So, yeah. Yes, sir.
So you had individual bees in the flow through chamber. Right?
Yes.
And I'm guessing at low temperatures, they probably just sat there, right? Because they're cold.
Yeah.
So then as you were testing this at warmer temperatures, did you try to quantify at all the bee activity? I'm just picturing a bee that's nervous running around that chamber, or are they just confined, they can't move?
They can't move. They're inside of a cocoon during this time. They spend from September, after post-pupal eclosion, they spend that whole time until the following spring inside of a cocoon, unable to move.
Oh, so you didn't have
I didn't dissect them
a
from the
bee like
cocoons.
this in a...
No. This is springtime when they're like this. This is after they've come out, chewed their way out of the cocoon and emerged, and yeah, they were still inside their cocoons, which made handling them very easy, actually. Yeah, definitely. You don't got them trying to crawl around on you. Yes?
So if I put up a mason bee house in my backyard,
Mm-hmm.
are these the kind of mason bees that I would get? Are they around here?
Yeah, most likely. Yeah, you would get a large number of Osmia lignaria. You'd also get some Megachile rotundata, which are the leafcutter bees. And you might get a few Osmia lignaria propinqua, which is a subspecies that I believe came from East Asia, although I may be mistaken. But yeah, essentially you're going to get mostly these guys right here, Osmia lignaria, say.
future climate models for this area, what do you predict is going to happen? Winters aren't going to be as cold.
Yeah. So, some have predicted that they will be able to weather this change, that they have enough plasticity in their development and in their wintering that they can, that a little bit of difference one way or the other, as far as when winter temperatures finally set in, won't have a large effect on their survivability to the following spring. It really depends on how severe of a climatic shift we're talking about, though. If we don't have any cold temperatures at all, then I would expect to see a pretty severe die-off with these bees. But
these are distributed, you said something about the same species in Utah, so are they throughout the West?
they are, so the Utah and Washington bees are phenotypically similar. And then there is a California population which has a slightly different development cycle. They emerge a little bit sooner and they go into winter a little bit sooner as well. The bees that we have, however, though, are really just limited to Washington, western Idaho, northwestern Idaho, and southern British Columbia. And then there, as far as I can tell, there seems to be kind of a break there before you link up with the Utah population, which seems to be a bit geographically isolated. Although I haven't seen anything to suggest that there is that they are from a common ancestor, although we could probably reason that that is the case. The California bees, on the other hand, are definitely different, having a totally different, in a common garden experiment that was done by Teresa Pittsinger, they found that the California bees were, they emerged sooner than the Utah bees did, even under the same thermal treatment. And yeah, they appear to be phenotypically dissimilar. Yes.
So related to that, one result I found really surprising was that I found such a big difference in the freezing point between the Washington and the Utah populations.
Mm-hmm.
And you said that was related, or it seemed to be related to body size. Is there a difference in mean body size between the two populations?
Ooh. I don't know. I don't know if body size was looked into in Sheffield 2008. I know that they generally have a pretty similar, they seem to have a pretty similar body size from the data that I've seen in previous studies across the spectrum. But they could be larger in Utah, potentially. But I'm not sure if that was actually looked into in that study. It's from one, the only supercooling data I was able to find was from one study that was conducted in Nova Scotia on bees from Utah that were sent to Nova Scotia to be tested. So, yeah.
What part of Utah?
Logan.
Oh, so that's going to be colder in the winter than here.
It is a little bit colder in the winter. Yeah. It's actually surprisingly pretty similar to what the temperatures that we see here in Ellensburg, though.
Okay.
But different from Puget Sound, of course. It's a lot more. It's the maritime climate over there on the west side, so it's a lot warmer, much milder. Yeah, it was surprising for me as well to find that they actually had a lower supercooling temp. Any other questions?
No other questions? All right. Thanks, Logan.
Awesome.